Hello everyone, I am Deepayan Rudra. Today I'm going to discuss about blasts, its types, and its algorithm in this video through this presentation. So first of all we will know what is blast, then we will see the algorithms of blast, and then we will discuss about the different types of blasts exists. So let's move to the first slide. So first we will know what is blast. So the full form is BLAST is Basic Local Alignment Search Tool. So BLAST basically stands for Basic Local Alignment Search Tool, the full form. Uh, it is a bioinformatics program which is used to compare a query sequence with sequences in a database. It identifies the region of local similarity. Generally, we will identify the local similarity through BLAST. So let's move to the second slide. Now we will know the purposes of BLAST. So first of all, we will use BLAST to identify similar sequences. Then we can use it for predict function of unknown genes or proteins of a query sequence. We can also use BLAST for study evolutionary relationships, and we can use BLAST to find homologous sequence also. So let's move to the next slide. So here we will see how BLAST work. Generally, actually this is the algorithm of BLAST basically. So first of all, we will take a query sequence, then we will break that sequences into a small words. Then we will search as the database for matches with that query sequence, and then we extends matches to form alignments, which is actually called the algorithm of BLAST. So here in schematic form, a schematic diagram, we will here it is discussed the whole process in a diagram also. So now we will move to the next slide. Now here we will see the different types of blast. So what are the different types of blasts? So there are major blast program, which is blast N, blast P, blast X, T blast N, and T blast X. So here through a diagram in a picture or image format, here it is here I present the different blast types. So now I will move to the next slide. So here we will discuss about the first type of blast, and which is blast N. So here what is actually blast N? A blast N generally compares nucleotide query sequence with a nucleotide database. Blast N generally used for DNA and RNA sequences. So some example of blast N is gene sequence comparison in bioinformatics. Now we'll move to the next slide, which is blast P. Now we will know this blast P is also a type of blast which we also already discussed previously. Now we will see what blast P actually is. So blast P, generally we use blast P to compare protein query sequence with the protein database. In case of blast N, there is nucleotide query sequence. Now here is blast P is protein query sequence with the protein database. We used for protein functions prediction. We generally use blast P for protein function prediction because it is done with the protein query sequence. So blast P is the most commonly used blast tool. It is very important point that blast P is the most commonly used blast tool among the all types of blast presence. Now we will move to the next slide, which is blast X. It is also a major type of blast. So here we will discuss about blast X. What is BLAST-X? Now, BLAST-X, we use BLAST-X to translate a nucleotide query sequence into a protein. So here the query sequence is a nucleotide, and we search that in a protein database where we use BLAST-X. Now we use BLAST-X to compare with protein database, and it is useful when protein is unknown. When we have an unknown protein, then we can use BLAST-X. Now we will move to the next slide, which is T-BLAST-N. It is also a type of BLAST, major type of BLAST. So what happens here? Now here a protein query sequence uh, versus a translated nucleotide database. So BLAST-N basically done between a protein query sequence and a translated database, nucleotide database. So T-BLAST-N generally helps to find genes encoding a protein. This is a use of T-BLAST-N. So here is a diagrammatic form about the different type of blast. Here I have discussed about the different type of blasts in a picture format, in a diagrammatic form. So now we'll move to the next slide, which is T-BLAST-X. Now T-BLAST-X, where we use, it's first of all, it is also a major type of blast. So where we use T-BLAST-X, a translated nucleotide query sequence versus a translated nucleotide database. There we use T-BLAST-X. T-BLAST-X is very sensitive. It's a very important point, and it is also computationally slow. So this is a very important point about T-BLAST-X, which is T-BLAST-X is computationally very slow. It is sensitive, but it is computationally slow. So now we will move to the next slide, and here we will discuss about the applications of blasts. So here are the different applications of blasts. So first of all, we apply blast to identify unknown genes and protein. Then we can use blast also to detect homologous sequence across species. We can use blast to predict a gene or protein function. So we can use blast to study evolutionary relationship, which is also called a phylogenetic relationship or phylogenetic tree analysis. So we can use BLAST to identify conserved domain and motifs. We can use BLAST to detect mutation, insertion, deletion in a sequence, in a query sequence. We can use BLAST to comparative analysis in genomics and proteomics. And we can also use BLAST to support in drug target discovery and disease research, which is very much important in today's world. Here, in a diagrammatic form, I have shown all the applications of BLAST here. So now we will move to the next slide, which is the conclusion. So the conclusion is BLAST is a very powerful tool in bioinformatics. So different BLAST types are used for different sequence comparison, and BLAST helps in gene identification and evolutionary studies. So that's are the conclusions. Then we will move to the next slide. So here I have given some references from where I prepared this PPT about BLAST. I have prepared this presentation about BLAST. So here are some references. So now we will move to the next slide.